YAHAWAH, BAHASHAM, YAHAWASHI, BAHASHAM, RAWCHAA, QADASH, double honors to GMS who told me this truth, Revelation 13:16, and he causeth all, both small and great, rich and poor, free and bond, to receive a mark in their right hand or in their foreheads. So we have an article here from Fast Company. This squishy material could be the next big step in computer brain implants. Researchers from Harvard and MIT say their hydrogel scaffold could be the secret to melding minds and machines. It may also help us learn more about how the brain works. So I'm just going to scroll down. So nearly all the known devices currently in development rely on tiny electrodes made from metal, not a particularly brain-friendly material. They pose the risk of infection and rejection, and because metal is hard and rigid, it can't truly conform to the contours of the brain, which makes it hard to maintain a secure, reliable connection with the organ's slippery surface. So it says, when we breathe, our brain has a little bit of micromotion. If you are gently shaking the bowl of jello with the spoon in its center, over time there will be a small spoon-induced cavity in the jello. In terms of a device, it means that there's tissue damage and scarring that occur over time. The soft brain interface described in December 2020 article consists of web-like scaffold made of hydrogel, a type of polymer material that can absorb large amounts of water without dissolving, like a very soft sponge. Hydrogels have the same mechanical properties and even the same mechanical character called viscoelasticity as the brain triangulates and can help reduce problems with rejection with rigid or stiff devices. One of the things that happens in any implant is the body builds a fibrotic scar to contain the implant and wall it off from the rest of the body since it's seen as a foreign object. Hydrogel electrodes shouldn't have that problem. So in the lab, the researchers seeded the hydrogel scaffold with human neural progenitor cells, blank slate cells that can transform into a variety of specific cell types, and after pulsing them with electrical current, discovered that they had formed networks of lattice-like structures on the scaffold and differentiated into multiple cell types with specific features. Some of the important types of brain cells discovered to be growing in the hydrogel, which include astrocysts, which aid in neural repair, and some of the word there, which creates a myelin sheath that insulates long, skinny nerve fibers called axons. Growing these axons has been an ongoing challenge in developing living models of the brain. So this is what they're working on. So the new hydrogel is not yet FDA approved, so any device incorporating it is likely 5 to 10 years from being used in humans. More immediately, says her colleagues plan to use the hydrogel scaffolds to create laboratory models of the brain that could shed light on the cell differentiation process and how, for example, neurons form connections with each other and other supporting cell types. Eventually, hopes these brain models could replace animal testing. Because the hydrogel really matches the properties of the brain, the cells grow in a really similar way to how they would in the body. We think this is the better place to test and then transition these drugs. Shalom.